reproduction types of bacteria include vegetative reproduction, asexual reproduction, and sexual reproduction. The first type of vegetative reproduction is the binary fission, and it is the commonest type of reproduction under favorable conditions in which cell divides into two similar daughter cells. And during this process, the bacterial chromosomes get attached to cell membrane and it replicates to the bacterial chromosomes. As the cell enlarges, the daughter chromosome gets separated. A crosswall is formed between the separating daughter chromosomes and it divides the cell into two daughter cells. Bacteria reproduce by binary fission, and in this process the bacterium, which is a single cell, divides into two identical daughter cells. It begins when the DNA of the bacterium divides into two replicates. The bacterial cell then elongates and splits into two daughter cells, each with identical DNA to the parent cell. Each daughter cell is a clone of the parent cell. Binary fission is similar in concept to the mitosis that happens in multicellular organisms such as plants and animals, but its purpose is different. When cells divide by mitosis in the body of a multicellular organisms, they cause the organism to grow larger, or they will replace the old worn-out cells with new ones. In the case of a bacterium, however, cell division isn't just a means of making more cells for the body. Instead, it's actually how bacteria reproduce or add more bacteria to the population. So now let's see what's happening genetically before the binary fission occurs. Here, the cell must copy its genetic material and segregate these copies to opposite ends of the cell. There are several types of proteins that comprise the cell division machinery assemble at the future division site, and a key component of this machinery is the protein FTSZ, and the protein monomers of FTSZ assembled into a ring-like structure at the center of a cell. Other components of the division apparatus then assemble at the FTSZ ring, and this machinery is positioned so that division splits the cytoplasm and does not damage DNA in the ongoing processes. As division occurs, the cytoplasm is cleaved into two, and in many bacteria, new cell wall is synthesized. Now let's see a few of the advantages of the binary fission. It only involves single parent and the offsprings are fully formed or there is no maturation needed. The process is very fast and almost 20 minutes is an ideal condition for it. After 24 hours, a single bacterium can give rise to 2 × 10⁶ kg of cells, which will be fair enough to cover the earth. And under disadvantages of binary fission, there is no genetic variety, or if it is faced with one unfavorable environmental condition, it can wipe out the entire population. Next kind of vegetative reproduction is budding, and in this case a small protuberance called bud develops at one end of the cell. This is followed by genome replication and one copy of the genome gets into the bud. Then the bud enlarges and eventually become a daughter cell and finally gets separated from the parent cell. Budding has been observed in some members of the planktomycetes, cyanobacteria, firmicutes and proteobacteria. Although budding has been extensively studied in the case of Saccharomyces cerevisiae, the molecular mechanism of bud formation in bacteria are not much known. A schematic representation of budding in planktomyces species is shown in the next slide. And in this diagram you can see the initiation of bud and bud formation and eventually the bud getting separated into a separate offspring. The next mode of bacterial reproduction is by sporulation. Sporulation refers to the formation of spores from vegetative cells during unfavorable environmental conditions. It may be described as an adaptive response that allows the organism to survive adverse conditions such as radiation, extreme heat or cold, lack of nutrition. Compared to vegetative cells, spores are multi-layered structures that tend to be dormant. These characteristics make it possible for some of the spores to preserve the genetic content of the organism during harsh environmental conditions. During certain unfavorable conditions, some of the vegetative cells go through a series of morphological changes that ultimately produces spores. A majority of spore-forming bacteria are gram-negative bacilli. These include aerobic bacillus and anaerobic Clostridium species. Some filamentous cocci have also been shown to be capable of sporulation. Depending upon the type of bacteria, four types of spores are produced. The first one is endospores. Endospores are the most common type of spores. They are typically produced by Clostridium, Sporosarzina, Bacillus, and the next one is a cyst. Cysts are a group of cells that join together in order to survive harsh environmental conditions. When conditions in the environment become unfavorable, the cells come together in a manner that allows them to survive. They are common in Azotobacter species. Unlike endospores, cysts are non-reproductive cells. They are also less resistant to tough environmental conditions and have a tough cell wall that is waterproof in place of the peptidoglycan layer found on the cell wall of bacterial endospores. The third type of spores are myxospores. They are produced by myxobacteria, and the myxospores are capable of surviving harsh environmental conditions such as UV light, high temperature, and desiccation. They are found within the fruiting body of the bacteria, and in contrast to the resting cells, they are formed from specialized spore-bearing structures. And the last type of spores are the exospores that are produced by members of the phylum Actinobacteria. These spores are produced through the budding of mycelium and can survive in harsh environmental conditions better than the vegetative cells. Sporulating bacteria are ubiquitous, means it can be found everywhere like in soil, water, and every part of the nature. Compared to the other types of spores, endospores are the most defensive structures capable of surviving harsher environmental conditions. And the reason for this is the fact that beneath the thin and delicate exosporium, they have a spore coat that is made of layers of protein. Another type of vegetative reproduction in bacteria is by fragmentation, and it occurs during unfavorable conditions. Bacterial protoplasm here undergoes compartmentalization and subsequent fragmentation, forming minute bodies called gonidia. Under favorable conditions, each gonidium grows into a new bacterium, and it becomes apparent that prior to fragmentation, the bacterial genome has to undergo repeated replication so that each fragment gets a copy. Next one is a sexual reproduction, and it occurs in the form of genetic recombination in bacteria. There are three methods of genetic recombination, and it includes transformation, conjugation, and transduction. Transformation was first studied by Griffith in Diplococcus pneumonia. In transformation, the DNA from the donor cell initiates heritable change in recipient cell. The transformation involves only one trait, though several may be acquired independently in this way. The next method is by conjugation, and it was first reported by Lederberg and Tatum in E. coli bacteria. Here, the transfer of genes occur by cell-to-cell contact. Cell-to-cell union occurs between two bacterial cells, and genetic material of one bacterial cell goes to another cell lengthwise through conjugation tube, which is formed by 6 spirae. As a result, the recipient cell possesses duplicate genes for a number of characters as seen in E. coli, which is an inhabitant of human intestine. The third method is by transduction, and it was reported by Zinter and Lederberg in Salmonella typhimurium. In this method, the genetic material of one bacterial cell goes to the other bacterial cell by agency of bacteriophages, or virus which is infecting the bacteria. In this image, you can see the bacteriophage and the host bacterial cell, which is the donor cell, and here you can see the recipient host cell. Here the phage will inject its DNA into the bacterial chromosome, and in the second stage the phage enzymes will degrade the host DNA, and phage with the donor DNA can be seen here. Here the cells synthesize new phages that incorporate phage DNA, and later in the recipient host cell we can see that the transducing phage, that is the phage with the donor DNA, injects the DNA, and in the final stage the donor DNA is incorporated into recipient's chromosome by recombination.